Greetings everyone, and today we are going to discuss about Phylum Porifera. And here it is basically an invertebrate, and as we know, it is a parazoid. Does not have any true tissue. It does not have any organ system, and it is aquatic. And as well as it is sessile. Apart from which, it is hermaphrodite, which means it's having both sex and both female and male uh, um, reproductive organs, reproductive gametes. Sorry, they are not having any sort of organ. They are having male and female reproductive gametes. Apart from which, they are sessile, aquatic, hermaphrodite, and apart from this, they are having their structure in such a manner as you can see over the screen. They are having their structure in such a manner that they are sessile and they attach on a rock, on a rock below water. On a rock below water, they always live in water. On a rock below water, these are mostly found in marine environment, marine water, while sometimes they are also found in fresh water. Apart from which, their size can range from, their width can range from a few millimeters. Width can be measured in a few millimeters, whereas its length can be measured over a meter. Length can be measured over a meter. So in that case, this is their size. Apart from which, they do not have any organ. And now let us discuss about how do they function. Basically, they are having their structure in such a manner that they are having uh, two layers of their body. They are literally, the word porifera means pori meaning pore, and fera meaning the bearer, which means they are pore bearer. They contain pores. They contain several pores which are known as ostium, ostia. Pores known as ostia, singular ostium. Apart from which, these ostia, pore ostium, and open into a large central opening, large cavity, which is known as large central cavity, which is known as ostia, which open into a large central cavity, which is known as spongocoel. Spongocoel, and that spongocoel is having water inside it in such a manner that here they are having several pores inside their structure, as you can see in the diagram. There are several pores inside their body. On their body wall there are several pores, and these pores are known as ostia, which open into a large central cavity, which is known as spongocoel. And what is the function of these pores here? They are of paramount importance over here, in such a manner that here these pores are nuclear pores. um, oops, these pores are involved in taking water. As they live in an aquatic environment, water comes uh, inside the body of a sponge through these pores, due to which, as the as the water comes inside the spongocoel through the ostia, the water is kept inside the ostia, and then uh, after being kept inside the ostia, the water current is then transferred out of the sponge through a large opening, which is known as osculum. Apart from which, water is taken from a large opening, which is known as osculum. Now when it comes to its structure, its line, its body structure, its wall structure, the sponge wall is having basically the following two layers, which includes the first outermost layer is known as a pinacoderm, which is made up of pinacocytes, whereas the innermost layer is known as gastroderm, which is made up of conocytes or collar cells. In that case, pinacoderm is made up of pinacocytes, um, and pinacocytes, as the name suggests, pinna means intake of liquid, cytes means cells which assist in intake of liquid are known as pinacocytes. Apart from which, these pinacocytes are having example. These are pinacocytes, and at the interval, as we can see, there are formation of several pores. As you can see, there is formation of several pores. This is a pore. This is a bit blurred, but here there is a formation of pore. This pore, ostium, is actually formed by dividing the cytoplasm of a special cell into two equal halves, due to which the cavity formed in between the cytoplasm of the special cell into two equal halves forms a pore or hole inside the cytoplasm, which is known as ostium, and that pore or hole is formed in the specialized cell, which is known as porocyte. Apart from which, when it comes to the second structure, there's an epidermal cell which is pinacocyte, and on the our innermost type, in the innermost cell, we are having conocytes. Conocytes are having an important structure which we're going to discuss in the next slide. Here, in between the innermost cell and the innermost layer and outermost layer, there is a layer which is present. Innermost layer and outermost layer, there is the presence of a non-cellular layer. There is a presence of non-cellular layer which is known as mesenchyme or mesoglia or mesohyl, and that mesoglia is not having, it's gelatinous in nature. Apart from which, that gelatinous nature is 80% in water, 20% in globular protein gelatin. Apart from which, this layer also contains some other cells known as amoebocytes, which are involved in amoeboid movement. Apart from which, good example of these amoeboid cells involves archaeocytes. Archaeocytes are cells, amoeboid cells, which are involved in amoeboid movement. These basically archaeoid cells are the totipotent cells, which are cells which are capable of giving rise to a complete organism, which is involved in the reproduction phase of sponges. That whenever asexual reproduction occurs in sponges, that is done by the archaeocytes through the process of budding or by the formation of gemicle, which we are going to discuss later. Now, this is how we are going to drive the structure in, and here this is the structure. Amoebocytes are present as well as for sexual reproduction. It's within the mesoglia. There is a presence of amoebocytes, as well as for sexual reproduction, and for asexual reproduction there is a presence of archaeocytes, and for sexual reproduction there is a presence of male gametes and female gametes in the mesoglia and mesohyl. Apart from which, when it comes to uh, the stability of sponge wall, the sponge wall is stabilized by the presence of endoskeleton. That endoskeleton is made up of two major components. What those two major components are? Those two major components involves first the spicule. That spicule is a rice-like structure, needle-like structure, which is composed of either calcium carbonate or silica, or either composed of calcium carbonate or silica, which collectively forms, which is composed of calcium carbonate or silica, which provides rigidity and support to the sponge wall, whereas another structure is present which is known as spongin fibers. Spongin fibers, being named as fibers, are fibrous proteins, proteinaceous in nature. They provide support by maintaining the shape of the sponge wall. They maintain the shape of the sponge wall, and spicules and spongin fibers collectively form the endoskeleton of sponge body. Apart from which, um, what else we are left with? We are left with um, some other structures. So this is a basic structure of how it actually occurs. Now let us discuss about the structure of conocyte. Conocyte is having its structure in such a manner that here, this is the main body, a soma of the conocytes. This is the amoebocyte, and over that, which is present in the mesoglia, mesohyl, and here there is conocyte, and that conocyte involves the main body, and attached to that main body are several microvilli. These are several microvilli attached to the main body, and those microvilli are having a mucus inside them. These are having several mucus inside the microvilli, and attached to that microvilli there is, inside the soma body, there is the flagellum which is attached to the um, conocyte. In that case, this is involved in, as flagellum being able to form independent movement causes to move water towards the osculum, through which the water is taken outside the body. Apart from which, when um, this is involved in an important procedure of transferring, of providing, of getting the food particles. So why is conocyte important? Conocyte is important because of uh, capturing the food particles, as well as it is involved in the process of capturing of food particles. As there are no specialized organs or tissues for it, there is the digestion, respiration, excretion, reproduction, all occurs by simple diffusion in the sponge body. How does this occur? So basically, as it lives in an aquatic environment, water enters inside the ostia, um, inside the body through the ostia into the spongocoel. Spongocoel, as water inside enters inside the spongia, as we know that we are having inside the structure of spongia, we are having spongocoel, we are having the conocytes, which is supposed to make the gastrodrums, or the inner layer of, which is supposed to make the gastrodrum or inner layer of sponge wall. Now in that case, the conocytes having, now as we can see within the conocytes we are having microvilli, over which we have the attachment of mucus. This mucus is then capable of catching the food particles in such a manner that passes through the when water passes through the mucus, when water passes through the microvilli, for suppose this is water is coming inside the uh, microvilli, the water is passing through the microvilli. As water is passing through the microvilli, it contains several food particles inside it, and that food particle goes inside the microvilli, due to which the food particle is attached with the mucus, and rest of the water is filtered out into the spongocoel, and from the spongocoel it is then transferred into the external environment by the osculum. It is transferred into the external environment through the osculum, from where the water is filtered out. And due to this, these are also known as filter feeders. Why these are known as filter feeders? Because they get their food out of the water by filtering the water out. So these are filter feeders because they get their food by filtering food out of the water. Now in that case, they get their food which is stick the towards the microvilli, and then the food is then taken inside the cell, and intracellular digestion occurs, which is that primitive form of digestion. Primitive intracellular digestion occurs, as well as there's no alimentary canal, as there's no connective tissues, or I mean there are no tissues, no true tissues at all. So there's no alimentary canal, so primitive or intracellular digestion occurs. Apart from which, we are having um, intracellular digestion inside conocytes. As intracellular digestion occurs, food is digested. Some of the food is assimilated within the conocyte, while rest of the food is then sent inside the mesoglium, mesohyl, where the amoebocytes perform amoeboid movement, due to which the food is then transferred towards the amoebocytes, as well as towards the pinacocytes, from towards the amoebocytes and towards the pinacocytes, towards the amoebocytes and towards the pinacocytes, towards the amoebocytes and towards the amoebocytes, towards the amoebocytes and towards the pinacocytes, towards the amoebocytes and towards the pinacocytes, by the movement of amoeba, which is amoeboid movement, due to which the food is taken inside amoebocytes and pinacocytes, and there they are able to utilize this food for ATP production, energy production. In that case, this is how digestion occurs, ingestion occurs, and intracellular digestion of course takes place, and the diffusion of food takes place simply through simple diffusion. Now in that case, this is how the digestion occurs. Secondly, we are having respiration, this complete system in which the digestion, in which the water is transferred inside the spongocoel through the ostia and then transferred outside the osculum, is known as canal system. It is known as a canal system, and that canal system assists in the following functions, which includes the digestion, as we have seen, respiration in such a manner that when water is brought inside the spongia, when water is brought inside the spongocoel, the water is brought inside the spongocoel, the oxygen present inside the water is taken inside the conocytes, while the carbon dioxide is released from the conocytes, due to which the gaseous exchange also takes place. Apart from which, excretion also occurs in such a manner that as they produce ammonia, as they produce ammonia, that ammonia is also released during the transfer of water into the spongocoel. When the water is brought inside the spongocoel, the ammonia is transferred from the conocytes and other neighboring cells into the water current. As the water current moves outside the osculum, the water current moves outside the osculum into the external environment, that causes the nitrogenous waste to be transferred out as well. When it comes to reproduction is here of two types. First, into sexual and asexual reproduction. Asexual reproduction involves two methods. First, it is fragmentation, in which there is formation of fragments which individually grow into a new organism, and secondly, there is budding and uh, a gemule formation, a gemule formation, budding or gemule formation. In such a manner, that budding or gemule formation occurs in such a manner. Budding and gemule formation occurs in such a manner that here budding and gemule formation occurs in such a manner that here a budding and gemule formation occurs in such a manner that here, as we know that it contains archaeocytes or totipotent cells. Totipotent cells. These archaeocytes or totipotent cells are basically amoebocytes, which are capable of giving rise to a complete cell. So, in the mesoglia, mesohyl, the archaeocytes form a network of. There is a formation of a network of archaeocytes, which is enclosed by the endoskeleton extension by slight extension of endoskeleton, which consists of the spicules, as well as which consists of the spicules as which consists of extension of endoskeleton including spicules as well as spongian fibers, which is enclosed which is supposed to enclose the archaeocyte network. In that case, that complete structure is considered to be an internal bud or gemule. That internal bud or gemule is kept inside the sponge, and whenever the sponge dies due to any external danger or external threat, whenever it dies due to any poisonous substance or external danger or any the unfavorable circumstance, the internal bud or gemule which is present inside the sponge comes out of the sponge, and then it grows individually as an offspring of that sponge. So this is how asexual reproduction works. And when it comes to sexual reproduction occurs in such a manner that they are hermaphrodites. Even though they are hermaphrodites, contain both of the sexes, they are they can perform cross fertilization, because if there's self fertilization, then then all sponges will have the same genetic makeup. In that sense, they perform cross fertilization, which is an internal fertilization, which means the fertilization will take place inside the body of that organism. This is internal fertilization. And cross fertilization such a manner that the canal system when water is taken inside the canal system through ostia into the spongo seal, and the sperms and the male gametes are released from the mesoglia into the spongocele, due to which they are transferred out of the sponge to the osculum and then transferred into another and the water is then intaken by another by another sponge ostia, due to which when water is intaken there, that male gametes, those male gametes fuse with the female gametes of another sponge, and thus cross fertilization, internal fertilization occurs. Apart from which, when it comes to the evolutionary relation, they are evolutionized by protists known as conoflagellates, known as conoflagellates. Hopefully, this all about this video. Hopefully, we'll meet again in another.